we get started, and if other people join, they will catch up. So welcome everyone to our webinar on how to optimize the refinery wastewater treatment operations with a biological approach. It is the webinar organized by EnviroZyme, the global leader of microbiological solutions for wastewater treatment plant operations. I am Evelyn Degentil, the commercial manager for the EMEA region. I am glad to see that today we welcome a lot of attendees from a lot of different places in the world. A couple of organization details before we start. So for confidentiality reasons, we just have hidden your personal information. Only Matt Livingstone, our global technical director here, and myself can see them. You are not able to speak or raise hands or make comments on the chat, but you can, throughout the webinar, ask questions or share some comments on the Q&A section on the top of your screen when you have the question mark next to the participant information. I will gather all the questions or comments that you have to ask Matt at the end of the presentation. I let now Matt introduce the topic for you.
All right, thanks Evelyn, and welcome everybody. I think most of the participants were designed today to be in Europe. We've done this in the US and North and South America. We'll do it again for folks in a couple weeks that are in Asia, adjusting the time a little bit, but I think we have people scattered in different places. So good morning, good afternoon, good evening, wherever you're watching from. This webinar is around biological issues that refineries face. And, you know, at EnviroZyme, when we look at troubleshooting wastewater treatment, we have this approach that we're calling Lead with Biology. And of course, that's our technology platform using bioaugmentation. But refineries are really a good example of where bioaugmentation can be useful. The wastewater contains a lot of complex chemistry that can cause issues at wastewater treatment, but a lot of these issues are suitable for biological treatment. So that's why we feel refineries are a good space, and we'll dive into some of the applications where bioaugmentation could be helpful in refineries. Want to do a brief introduction to EnviroZyme because I think there's some folks that may not be very familiar with who we are and what we do, and we'll do a brief introduction to the bioaugmentation technology. As Evelyn mentioned, we're really the world leader in bioaugmentation. We're a subsidiary of the Betco Corporation that's headquartered in Bowling Green, Ohio. That's where we also have our production. It's not far from Detroit, about an hour south of Detroit. And at our fermentation production facility in the upper right-hand corner, we ferment bacteria, we blend them together, we package both custom and off-the-shelf microbial solutions. We have local and technical support covering North America, Latin America, Europe, and Asia Pacific. So we truly cover the entire globe. And we bring products out to market through partners, through both local partners that may be in a specific region and through a few global partners that cover wider regions. In Q1 2026, in the next couple of months, it's going to be pretty exciting. We're expanding our production. So you can see in the upper right-hand side, the lower left of that picture is all new space. So we've grown our business and have invested in the business, and we're doubling our production capacity. So that's pretty exciting to kick off 2026. EnviroZyme is really the combination of a couple different companies historically that were in the bioaugmentation space. It started in 2009 when Betco acquired Biosystems International. And then in 2023, we acquired the wastewater business from EnviroZyme, which is now Novonesis. And then in 2024, acquired the business of BioWish Technologies. And rolling those companies up really makes us the largest player in this wastewater bioaugmentation space. What we do in our production facilities, we take organisms that are found in nature and they're organisms that we think have a unique capability or are well suited for wastewater treatment. We'll grow them up first in a small media flask and then into a larger seed fermenter through the production process into larger 30,000 liter tanks. We'll grow those bacteria up at a very high concentration, centrifuge them even further. Some of the strains will spray dry, and then we can, through a number of different production technologies, either finish that off as a dry product or as a liquid product or as a solid product. And we're not going to get too much into those different product formats, but just a quick overview of our production capabilities. When we talk about bioaugmentation in wastewater treatment, it's really we define it as the practice of enhancing the performance of the indigenous bacterial population through the addition of concentrated natural bacterial strains with specific abilities. And we look at typically 4 main applications of this. And if you think of that over time, the first would be the startup, and that's where we're reducing the time for a wastewater treatment plant to come online, to become commissioned and reach its full capacity. So in this example, we have a solid line and the dash line. The idea is that with bioaugmentation, you get to that startup, get through that startup period faster. In some applications, there's an efficiency improvement. So we're looking at improving the overall plant efficiency around COD capabilities, degradation capabilities. That's often with existing equipment. A lot of times a plant might be undersized if the factory was made 30 years ago and the factory's expanded and the wastewater treatment plant stayed the same. It's going to be undersized, and sometimes bioaugmentation can help increase that efficiency of the plant. Where we get into applications in refining is more around stability and upset recovery. So stability is in a wastewater treatment system. You're going to get influent that's going to be changing. It's going to be variable, especially in a refinery. You might have some harsh chemistries coming through that can upset the wastewater treatment plant. The idea is to maintain some sort of stability so that you're not going through an upset. And then the upset recovery application is simply that. So if you get an upset and you lose some treatment capacity or some of the bacteria die off in your system, the idea, similar to a startup, is that you use bioaugmentation to get back up to that treatment efficiency as quickly as possible. So we kind of look at those 4 main applications. I'm going to talk further on why refineries are good candidates for bioaugmentation. Crude oil is a mixture of a lot of different hydrocarbons, but also other components, and that varies by deposit, where it is, and what's in the location of that deposit. And large oil companies are scanning the globe all over to find new oil reserves. And those reserves are going to differ. Some of the cheap crudes are easier to get at, and some are cheaper and more abundant. And I would say in the last 20 years or so, refineries have really upgraded to be able to handle some of those crudes that are of lower quality. When we talk about sour crudes, it's talking about having a higher sulfur content, so between 0.5% and 5% sulfur. So there's more sulfur to remove in the process. Talk about heavier crudes as being more viscous. Those are generally easier to find from an exploration standpoint, but can cause impacts in a refinery, especially switching between different crude slates. So that recently, I would say in the last 10, 15 years, has really been the driver for a lot of the issues at wastewater treatment plants. So when we think about who's refining sour crudes, at some point it was not very many. Most refineries were getting sweet crudes, but as the technologies enabled refineries to do more processing, it's really all the large players around the world are seeing and treating heavier and more sour crudes. So, regardless of the region where you are in the world, that's a trend that's everywhere. What I wanted to try to do is take a quick survey of the attendees here. And I'm going to switch my screen to a Menti poll that hopefully you'll be able to see. And if you could scan that with your phone, I think it should be up, just to get a sense of is your refinery, or if you're working with refinery customers, do they process sour crudes or opportunity crude feedstocks? So if you could take 20 seconds or so, scan that up, and maybe we'll see what most folks are dealing with. And I would say if there is an issue with that, maybe just plug it in the Q&A and we can address that later on. This is the first time we've tried to do this, so bear with us if you're unable. All right, I'm going to move. Well, here we go. Got a couple of those. So periodically run sour crudes, a couple more. Okay, so most places are seeing some periodically run sour crudes, and that's interesting because that means that the refineries are switching back and forth, and we'll get to what kind of issues that can cover. So let me close out of that. I'm going to go back to the PowerPoint. Thank you for those who participated.
Please, you can keep that on your phone. He will do other polls later on that you can you won't have to scan it again.
Yeah, just keep it open. We'll drive to that in future. Okay, so in a refinery, they're very complex. I'm sure most of you have been there, or if you're at a refinery, there are a variety of different refinery units depending on what they're treating and what they're producing. But all these key contaminants come down, are combined or removed from the crude in some way, and then combined in the wastewater treatment. And while they're higher toxic at higher concentrations, most of these compounds are degradable and can be managed through the biological treatment process. And that's what the treatment plants are designed for. Now, upstream of the biological treatment, there's pre-treatment. And pre-treatment is really important in protecting and preserving the wastewater treatment. So it could be an API separator or a DAF or equalization. These can be in a different flow order. You can have EQ up front, but it is worth optimizing, investing, and maintaining pre-treatment because, again, that's really the last defense for taking things out before they get into the biological treatment system. And a lot of issues can be minimized by simply improving pre-treatment. So when we get into the biological treatment system at a refinery or any wastewater treatment plant, we can break it down to very simple terms in that the bacteria are there to remove pollutants from the water. So that's how they grow and survive, is on the organics and the nutrients and pollutants that are in the water. And then bacteria form a dense flock, and that's through the wastewater treatment process, the way the processes are designed through aeration and clarification for the bacteria to flocculate. And ideally, you get a nice dense flock, like this flock here, that's going to settle out, and you have a very clear effluent that goes over the weir, and then you can return some of this sludge or dewater some of that sludge efficiently. And that's the ideal situation, although it doesn't always happen that way. There's a lot of pressures and things that can influence how well bacteria flocculate. That can be competition among different bacteria, it can be competition or predators from higher life forms in the wastewater. It can be toxins, so toxicity, heavy metals, high concentrations of ammonia or phenol that come through can be toxic to wastewater, toxic to bacteria. That can impact the flock. The sludge retention time, how long the sludge stays in the system, how old or young the sludge is. At a refinery, you often get pH swings or variable pH that comes in. That can be a stressor. The substrate, the organic substrates, dissolved oxygen concentration, or even the ability for dissolved oxygen to get into the flock. So the oxygen transfer is an important aspect of getting a healthy flock. Temperature, it's often in the higher temperatures. I was in some refineries early this year in Korea. They're seeing temperatures up in the high 40s, and that's going to impact wastewater treatment. That's going to cause stress on the biomass, and that's going to impact certain processes. And then inert material. Also, it's not just bacteria that make up the flock. There's also inert material that comes in that becomes part of this flock. And that can also disturb oxygen transfer, or other issues with flocculation. And what happens is you have a person in a control booth, you have the operator that's trying to manage all these issues around trying to form a good flock and trying to get good treatment. And that's key, is really understanding what these growth pressures are, how they're impacting your system, so you can manage it in a way to minimize that and try to get good, healthy flock. So I'm going to try to do another poll, and I'm going to slide this ahead so it should be up on your screen. And I'm going to move here so we can share the results and see. This one is around the most persistent biological issues at a plant, and you'll have some choices there that you can find. All right, so seeing some things come up around filamentous bulking and settling issues. Yeah, we'll talk briefly about that later on. All right, we'll go back, and you can answer those during the presentation. We'll get back to this at the end and just kind of refresh on some of those results, but I'll get back to the presentation. So another vote came in for toxicity-related upsets. So then we'll get into kind of troubleshooting approaches and good ways to think about identifying problems and managing issues at a refinery. The first level is kind of going in and doing a site visit and doing data review. It's really important to get on site to, understand the system, not only looking at it at a flow diagram, but seeing it in person and seeing those processes in real time. Getting the right data and reviewing the data. So if someone's describing a problem, is the plan, or are you able to review data that accurately describes the problem? So if you have a problem and the refinery is saying, hey, we've got a settling issue, then you should be able to see that in either the SV30 issue or SV30 data, or SVIs if they're calculating sludge volume index. If it's a pH issue, you should be able to measure that and find those things. So you want to be able to make sure you're able to review data and review the right data. I'll make a short pitch here for some of the AI tools that are now available. It's becoming easier and easier to upload data into some of the AI platforms, where they can very quickly and with a lot of data do trending analysis and look for ways you can do certain prompts to have it scan data in certain ways. And I'm finding that to be very helpful. I think we're really on the edge of that. We're seeing it more and more, and curious if other folks are doing that as well. But just wanted to put a shout out that for data analysis, again, really important. The next level is we look at is looking at looking microscopically at the biomass, and there's a lot you can tell sometimes that you can't see through the data, through managed data through a spreadsheet. So what do we look at when we do a micro exam? It's really four main components. One is looking at the flock. So again, do you have a dense flock, a flock that's going to settle, a flock that doesn't have a lot of EPS on it? We'll talk about EPS in a little bit. We also want to look at the filaments. So not only how many filaments are there. This is a pretty healthy flock particle here that you see. You see some filaments standing out, but not too many. So you want to get a filament abundance. But also, if there are a high amount of filaments, you want to be able to identify the filaments. So you can look at filaments under high magnification, and through some morphology assessments, identify the different types of filaments that you have in the system. You can look at higher life forms, or some of these single cell or multicellular higher life forms that are in there. They can be indicative of the conditions, whether healthy or toxic conditions, or how old the sludge is. This Suctoria organism here is a good indicator for nitrification. You often see them when there's good nitrification, or if you see them, it's often a good sign. And then the last is looking at the bulk water. So looking around the flock and looking in the spaces in the water. Is there a lot of free bacteria, or is there a lot of inert material? So those are the four main components we look at microscopically to get a good assessment on the biomass. And we have a lot more information. We may do other webinars here in terms of how to do these micro exams. And then the third level, again, is fairly new, I would say, and becoming more accessible all the time. So this is using DNA analysis and microbial community analysis in a wastewater setting. This technology started to become available, I would say, 10 years ago. And the issue is that you could get a lot of information. It would take a long time. And it was really hard to understand, what do you do with 200 or 300 different bacteria that are really hard to, categorize and understand. But in the last 10 years, there's been a lot of work around databases and understanding what the different genuses can do. Typically, we go down to the genus level when we look at DNA analysis. We use a partner called AsterBio. They're based in the US. There are other companies similar that do this kind of work. But what's nice is that some of this information now is that you might get a couple hundred different genuses that you can look at, but they're separated into different functional groups like ammonia oxidizing bacteria, nitrogen oxidizing bacteria, nitrogen reducing bacteria, down to foaming bacteria, filamentous. So you get a good sense of not only what's in the system, but what the function is. So if you're nitrifying, if you have a plant and you're very concerned about nitrification, you might want to take a sample when things are going good to get a sense of what percent of the DNA in my biomass, what percent of those organisms are ammonia oxidizers or nitrifying bacteria. And that's a good way to kind of get a baseline. And then when you come into issues, you can do another test and see how it changes over time. And this is a really good way, and I think will become more and more important to understanding and improving wastewater treatment performance. I would say also this is another really good use of AI tools and being able to load this in and be able to run through a couple hundred genera and look for specific capabilities. And some of those platforms can very quickly also put these into functional groups. So that's been an advancement as well using AI. So in a refinery, before we get into some of the issues, we talked about these 4 applications for bioaugmentation. Specifically in a refinery, it's looking at either a startup or more frequently recommissioning after a turnaround. So after some maintenance is done as a refinery, it's going to get back up and running. That's kind of a startup application. System efficiency, often around when there's higher low temperatures, looking for BOD or COD improvement, nitrification or TSS or better settling, removing specific hydrocarbons or phenol. That can be in the system efficiency. Instability, again, getting variability from the feed, changing crude slates or toxicity, settling issues, different sorts of stress. And then upset recovery is often around nitrification, or if there's a toxic situation or hydraulic washout, that would be more along the upset application in refineries. So now specifically down to some of the issues in wastewater treatment. I'm going to start talking around EPS, and that's extracellular polymeric substances. And we'll show some pictures and get into a little bit more of what that is. This is really the most common biological problem globally in refineries that we see when we're working with customers. And again, this is a microexam image. This is an image using India ink or carbon black ink. And you can see that this is a flock particle that has a lot of EPS or slime bulking, and that black ink material does not penetrate. It penetrates into the normal flock bacteria, but doesn't penetrate through the slime. And you can see here it stands out. They have a flock particle that's mostly made up or covered in slime. Or this EPS material. EPS consists of polysaccharides or proteins or lipids and DNA, so it's a complex mixture that varies among different bacteria and often different situations. So different stress events will change the composition of the EPS. And there's a whole wing of study around EPS in microbiology. And it's visible with different ink stains or just through phase contrast microscopy. And the reason you get bulking or slime bulking is that it's a protective measure for bacteria, and we'll talk about that in the next couple slides. But first, I want to do a quick poll, so I'm going to move the poll forward to EPS, and just wondering if people know about EPS or at your facility, or if you go on site, how confident you are in identifying EPS. And I guess I'll toggle back over there. We can take a look. So someone's somewhat confident in identifying EPS, not very confident. Again, it's one of those issues with a microscope, not very common. It becomes one of those things where a microscope really helps in the identification. And we'll talk about some of those ways to identify in just a moment. All right, I'm going to hop back over. So EPS, there are a couple different types of EPS. The first one we're going to call tightly bound EPS. And that's where the EPS is close to the cell. It's almost on an individual cell basis. I'm going to blow up this fraction of the microexam. This flock here is mostly normal flock, but you can see these bacteria on this side look a little different and blown up. You can see, and even here on this kind of diagram, you have the bacterial cell in the middle, but this slime layer coats each of these bacteria, and they're kind of equal distant apart from each other. And what tightly bound is caused often by high FM. So when you get a readily degradable substrate, often there's so much food that the bacteria can't process it all at once. So it kind of stores it up as this kind of EPS material. And that's a result of a slug load of degradable material. It can also come from a nutrient deficiency where you might not have enough nitrogen or phosphorus to process all the food that's coming in. So the bacteria, again, stores it up, and that's a survival mechanism for the bacteria. And we'll talk about some of the problems that causes in a little bit. Then there's also loosely bound, and that's more of a slimy kind of matrix. I'll move it forward here. Again, most of this is kind of a flock that's not super dense, but this area here, this kind of area, you can see where it's blown up, that there's a lot of kind of gel. It almost looks like a gelatin matrix here, and you have the bacteria. And it's not so equal distance like the other one. This is just a big blob of slime where you have bacteria kind of hanging out in here. And this loosely bound EPS is caused by stress. It's storing up reserves like the other one, but it also provides a protective measure from toxins and environmental changes. So when there's a big change to the water environment, if bacteria form this kind of slime and aggregate together, it's a protective environment for the bacteria that are on the inside. We often see this where there's a longer sludge age, at least 10 or 20 days, but sometimes longer, is where you find this loosely bound EPS. Now, the issues with EPS is that, one, that it's not very dense, so it doesn't settle very well. When it does settle, it doesn't compact very well, and it doesn't dewater very well because there's a lot of water bound up into that gel and into that slime matrix, and it just doesn't come out through standard dewatering processes. So the chemical use goes up, sludge disposal cost goes up, so you're disposing more sludge. And another issue with EPS that we're seeing is that we'll often lose nitrification because nitrifying bacteria like to form and hang out on dense flocks that settle in the clarifier and return back into the system. And if the flock is not, is it washed out, or if it's not a good place for nitrifiers to kind of survive into, then plants will often lose nitrification. Also, you see a lot of foam and scum on the DAF or in the aeration basin or in the clarifiers. So you often have a TSS issue where you have suspended solids going out. And it also costs money to send somebody out there to remove scum or remove foam off the top of a clarifier or aeration basin. So it's causing money that way. And then it can also cause oxygen transfer issues. So oxygen doesn't transfer very well through that material when it's tied up in the flock. It can block oxygen from getting inside the flock. So again, often see some ammonia issues, nitrification issues, BOD, COD issues, and then sometimes even whole effluent toxicity issues if you're not getting good oxygen transfer. And again, that can be tied to the EPS. So again, it's kind of a hidden issue. It's not often observable with your naked eye. If it gets bad enough, like in this situation here, you can see it on top of a clarifier, but sometimes you don't see it, and that can cause settling problems that are hard to diagnose if you're not looking at your sludge under a microscope or not sending it off for a microscopic analysis. So possible causes for EPS, loading and feed variability is key. Increase in organic loading or inorganic loading, or if there's a change in the crude. Earlier in that polls, a couple places were saying that they, sometimes or occasionally take sour crudes in. That change can really impact wastewater treatment. Or if there's an unknown chemical dump in the system, that variability can cause stress. Nutrient and chemical imbalances, typically we look at like 105 to 1 between BOD and nitrogen and phosphorus. If there's not enough nutrients in the system, that can cause EPS, pH swings. If the refinery or pretreatment is trying a new chemical, sometimes that can cause stress, seasonal variations, or operational. So if there's a process shutdown or aeration or mixing issues, if there are issues with with aeration or in the manifold for distributing air or mixing, that can cause issues, operational changes, equipment problems, if pumps aren't working the right way. So a lot of things can cause stress, and that stress can cause EPS. Approaches to treating EPS, again, it can only be removed by wasting. So there's no bacteria that's going to go into the system and chew it up or degrade that gel material. The best way, the only way to get it out of the system is by wasting. So by increasing wasting, we get it out of the system. Bioaugmentation can be helpful in building back a healthy biomass and handle readily degradable shock loads, especially for tightly bound where there's shock loading issues. So bioaugmentation can help fend off tightly bound issues. And then another thing, if the plant has the capability to introduce step feeding to distribute BOD load across the aeration basin. In some places where the BOD comes in all in the same place, that can cause an area where there's a very high F to M just in that microenvironment around the influent. And sometimes when that influent is spread out across the basin, that can help solve some of the tightly bound EPS issues. Similar with loosely bound issues, loosely bound EPS, you want to identify the causative factors so you can make a change to reduce it. It's not just wasting it out isn't going to be enough, because once you stop wasting or get the sludge back to that same level, very likely the same issues are going to come through. So do some detective work, look for ways where stress might be coming into the system, waste out the EPS. Once you waste it out, bioaugmentation can be helpful for building back a healthy biomass and improving stability, fending off some of the toxic compounds like phenol, amines, and help minimize the stress in the system. Typically in a refinery situation, the product that we would recommend is BioRemove HC. Some of our distributors might call that product something different, but it's a product that's designed around hydrocarbons, handling a variety of different hydrocarbons like phenol and longer chain hydrocarbons. That's typically what we'll use in a refinery application. So EPS, just in summary again, it's the most common issue that we see, the most common biological issue that we see in refineries processing sour crudes, which again is most refineries, leads to high chemical costs, can lead to discharge violations. It's hard to manage the issue if you're unaware that you have EPS, and if you're unaware of how to identify it. So it's important to either have a microscope on site or be able to send out the sludge for analysis. Again, tightly bound EPS caused by readily degradable BOD can be managed with bioaugmentation and maintaining a lower food to mass ratio. Loosely bound EPS, there are multiple causes, but usually manageable through operational control. Bioaugmentation can help stability, and then proper identification of the causative factors so you can manage that better through operations. All right, so that's a little bit on EPS. want to talk about nitrification. Yeah, we're doing pretty good on time. Nitrification, typically look at the nitrogen cycle, and nitrogen moves through the environment, through the atmosphere, through plants and animals, and through the soil and through water. This is all natural processes that are mitigated by bacteria. And in a wastewater treatment plant, it's the same thing. It's leveraging different processes that different bacteria have, and those are engineered into the wastewater treatment system. So you have a plant that can take in nitrogen in a variety of different forms and remove it completely from the system through total nitrogen, through either nitrification and denitrification. We'll talk about that process, but I'm going to get to our last poll. I'm going to move it on to the next one. So if you look at your poll again, has your facility experienced nitrification failure in the past 12 months? And there's a couple options on that. I'll share the screen so we can see some of the results come in. And this will be the last one. So in the last 12 months, if you've seen stable nitrification or if you've had nitrification disrupted. Okay, once or twice. Nope, came close. A couple once or twice. All right. Well, good. I'll hop back over. And again, nitrification is a common issue that we see a lot of refineries. And again, an issue that's well suited for bioaugmentation. We'll talk about some of those applications. All right, so in a wastewater environment, we showed that the quick nitrogen cycle in nature, and we didn't want to take a lot of time to go through that. But in a wastewater treatment system, you've kind of got four main processes. You have ammonification. So if nitrogen comes into the influent in amines, or it might be in terms of protein, maybe not so much in protein in a wastewater, in a refinery situation, more frequently amines. But if you're doing food processing or something else, it may come in higher in protein. Bacteria can convert those nitrogen, those amine groups, into ammonia, and that's mitigated by bacteria. The second process is assimilation, and we talk about bacteria needing nutrients. That's that 105-to-1 ratio, where for every 100 parts of BOD, you're going to need 5 parts of ammonia nitrogen. And that nitrogen goes to creating proteins in bacteria or DNA, a lot of different processes in a bacteria, or a lot of the building blocks require nitrogen for those bacteria to grow and replicate. So bacteria just take that up and incorporate it into the cell. Another way ammonia is converted is through nitrification process. And this is kind of the classical two-step nitrification process where you have ammonia oxidizing bacteria, converting ammonia to nitrite. And then a second stage set of bacteria, the nitrogen oxidizing, nitrite oxidizing bacteria, converting nitrite to nitrate. And then if your system and plant is set up properly and it's operating properly, you can do a total nitrogen removal, a denitrification process where you convert nitrate to nitrogen gas, and that nitrogen gas bubbles up out into the environment. So those are the four different processes that, again, are simultaneously happening in a wastewater treatment plant. When we get to nitrification, this is really key, and this is a key slide for nitrification. We kind of look at this checklist. Nitrifying bacteria grow very slowly. So compared to the bacteria that degrade BOD, in a regular environment, those bacteria in a treatment environment might be multiplying every hour or so. In a production environment, even every 20 minutes or so. But in a wastewater treatment environment, nitrifying bacteria are going to double once about every 24 hours. So they grow much more slowly. So if they're impacted, it takes them longer to recover. And that's key to why nitrification, again, is a more sensitive process than COD removal. If you have something toxic that comes in, the first thing that's going to be impacted is nitrification. And nitrification, I'll just go through this kind of checklist. From a pH standpoint, The ideal range for nitrifying bacteria is in the upper 7s, closer to 8, 7.7, 7.8. But in that 7 to 8 range is really ideal. But you're going to experience trouble if you're below 6.5 or if you're higher than 8.5. And again, this is rule of thumb. I've been to plants that nitrify below 6.5, but that plant is, set up to run that way. In a normal situation, it's usually between 7 and 8. DO, typically above 2, requires a lot of oxygen to nitrify. And in a refinery application, sometimes even higher if you have a lot of inerts or other issues that are causing oxygen transfer issues. Typically above 2.0. Gets below 2, get a little concerned. Temperature ideally in the high 20s, 20 to 30 C. When water gets below 15 or above 35, that's where issues start to come into play. Above 40 is where nitrification will often start to crash in a refinery. And I think in refineries, it's typically higher temperature issue concerns than the lower temperature side. Alkalinity, for every kilogram of ammonia, you're going to be required to use 7.14 kilograms of alkalinity as calcium carbonate. So you need alkalinity in the nitrification process. Typically, we look for a residual of 50 milligrams per liter. If there's no or a lower residual than that in the effluent, we're going to look at that as being a possible issue. You'll also start to see some pH issues if you're not adequately buffered or if you have alkalinity that's too low. Phosphorus, we're looking for residual in the effluent, so anything, more than 0.1 or 1 ppm. If there's no residual in the effluent, we want to go back and look at adding phosphorus. We've seen that happen. Sometimes people don't want to have any phosphorus in their effluent. They run out of phosphorus, and that impacts nitrification. Typically looking for a BOD less than 50. Again, BOD and COD degrading bacteria are also consuming oxygen. And since they grow faster, they're going to outcompete nitrification. All things being equal, they're going to outcompete nitrifiers. So we want to get that BOD down below 50. And the sludge age, again, in a refinery, some municipal plants can run at a much lower sludge age, but in a refinery, typically we're looking above 10 days. Some refineries can get away with a lower sludge age. So if you're lower than that, you might be putting your system under stress. We see a lot of refineries sometimes going with very, very long sludge ages, longer than 30 days. Sometimes what can happen is you get toxicity built up into the sludge. I'll give you an example of that in a little bit. That's something to watch out for. And then, again, you want to have a healthy flock. You want to make sure you have flock in the system that's going to settle, that provides a good nesting site for nitrifying bacteria. If they're just out floating around on their own in the system, they're not going to settle, and they're just going to get washed out of the system. So nitrifying bacteria like to form on a healthy flock. So that's one thing we always want to look for is there a healthy flock in the system? Okay, so bioaugmentation is one of the most frequent applications that we come across, especially in refineries and wastewater treatment. So when to consider bioaugmentation as a solution? One would be when there's frequent upsets, when there's a slow recovery. If you have an upset and it typically recovers in a day, 1 or 2 days, it's not so big of an issue. But if you're recovering, if it takes a week, some places we see frequently it takes 2 weeks or 3 weeks to recover. That's a good application for bioaugmentation to speed up that recovery. Again, if it's an overnight recovery situation, then your system is pretty robust. But if it's longer than a couple days, it's a good application. If your plant has a history of nitrification, but it's unable to recover, if there's an upset and you just can't figure out what's wrong, you're going to have to do some troubleshooting. But if you can figure out what some of the issues are, it's a good application to get it up and running. If you're at a plant that has no history of nitrification, you want to do some homework first. It's not so simple to just add nitrifying bacteria to expect the plant to nitrify. So a couple different applications that we'll talk through a little bit. If a plant has, I'll get through this and then we'll still have a couple minutes for questions. If a plant has a slow recovery. As we talked about, recovery taking more than 1 or 2 weeks. That's a good application. I'm going to show an example where at a plant, this is real data, where a plant lost nitrification for 20 days. And this would have been a good, we can't go back in time, but it would have been a good application for nitrification. I'm going to share some of the data that they have. We went through this nitrogen checklist. And just for the interest of time, I'm going to move this forward to show that the issue was caused by a pH swing. It was a pH swing sometime in June of that year. You also saw that reflected where the alkalinity was lowered because that was taken up by this lower pH. We looked at COD, phenol temperature, no other issues. It's just a pH swing that was causing the issues. And again, it took about 2 weeks for that to come back in. I want to share kind of a theoretical application around bioaugmentation where we're looking now at the ammonia of what happened at the plant. This is the pH. We saw the pH drop down. If we were adding nitrification, we would normally see once the pH starts to get back into that normal area. Once it's over 6, we start to see a good recovery. Typically, we would, often see a recovery like this in about 7 days, shortening that time for about 14 days. So that's kind of an example of where we see bioaugmentation shortening that time frame. If it's a plant that is not nitrified, what's changed? Has a crude changed? Is there a new process? You want to go through the recovery checklist, look at the micro exam for stress indicators, potentially do the community analysis and say, hey, if we know if your plant nitrifies well and you've got ammonia oxidizers at 1.2% or 1.9% of the population, take a look at the system and see if the nitrifier is there or if they're not. That's often a good way to do some troubleshooting. And the last thing around nitrification is doing a toxicity screen. Sometimes a crude test like this on a benchtop can be pretty quickly to run. We've got this procedure. If anybody wants it, you can email myself or Evelyn. We can forward that to you. In this case, this is a plant that had a 35-day sludge age. Nitrification was happening. It slowly was deteriorating, and then they lost nitrification, and they couldn't get it back, and they were suspecting toxicity. So we recommended a quick and dirty test to be done at the refinery. We asked them to get a beaker with 100% influent. They had about 50 ppm of ammonia in the influent. We split it between mixed liquor and influent, and then in the last beaker, we just put mixed liquor in. For all of these, we adjusted the ammonia, so the ammonia was the same concentration. And what happened was that the influent nitrified overnight. This is the first couple hours of the day they set up the test. Overnight, it was fully nitrified. The mix between mixed liquor and ammonia was mostly nitrified, and then the mixed liquor itself was not. And what was happening was that there was something toxic built up in the mixed liquor, stuck into the flock particle that was causing toxicity. The recommendation was to improve wasting, to use BioRemove HC to replace some of the wasted sludge to help degrade some of the toxic compounds, and then BioRemove AM, the nitrifying bacteria, once they repeated the test and found that the mixed liquor was positive and not inhibitory anymore. So that's one way, again, to use kind of a quick and dirty test. Nitrification, again, the most common application, go through the checklist, do a micro exam, look for stress indicators. And I'm going to jump ahead to the last one. Don't add nitrifiers to the system if the system isn't ready to nitrify. So if there's some items outside of that checklist, it's just not going to happen even if you put nitrifying bacteria in. So we want to make sure that the system is ready to accommodate nitrification. All right, we saw earlier there were a couple plants that talked about sludging, filamentous bulking. We're going to do another webinar in the future. Quickly, the idea is that filamentous are bacteria that grow end on end. They cause issues because they cause issues in settling. They cause issues compaction. If you can identify the bacteria, they're often put into buckets for different causes. There are about 20 or 25 well-known bacteria that are caused by one of these 6 or 7 issues around low food to mass ratio, low DO, septicity, substrate related, low pH, nutrient deficiency, or oil and grease. If you can identify the filament and identify these conditions, it's a really good way to kind of jumpstart the troubleshooting. Then you get into the data and you look, okay, if you think it's a low pH, then you look at your data and say, are there times where the pH is low, and can you correlate that to pore settling? That's how all this works together in terms of a data review and a micro exam. So I'm going to skip forward on that one. Some other issues around, often you see weak flock or pore settling due to an older sludge. I'm just going to skip through this one, again, just in the interest of time. Some other issues that happen in refineries that we weren't planning on addressing today, but just differences in loading, whether you have a degradable shock load that's biodegradable or recalcitrant COD that's harder to degrade. TDS, having high TDS come in, or H2S control, toxicity, oxygen deficiency. All these are areas that we can further explore. I'll also run through some of the applications that we use, the nitrifying bacteria, the bacteria that are good for hydrocarbon degrading. We have another product that's frequently used in startups or restart situations, upset recovery, called BioQuick 5140. It has a small inert material that bacteria grow very well on, so it settles out very well. It stays in the system, can help you return settled bacteria into the system, and some other products for either sulfide control or nutrients that you can contact us, get into more information on. Here's our global team across North America, across Latin America, in Europe, and Asia Pacific. So you can reach out to your account managers or business managers in those regions. So thank you for sitting through. We've got 5 minutes left for questions. So Evelyn, I'll open it back up to you. If anything's come back in, feel free to share here for the next 5 minutes.
Yeah, do not hesitate to type questions. Matt, one here for you while waiting for potential other ones. Can you explain a little bit on how the microbial analysis, the microscopic analysis, can help troubleshoot some issues that you mentioned into your slides?
Yeah, I think the key is when things are running well to benchmark your system. So if nitrifying, if we're talking about in particular, nitrifying doing a quick DNA analysis and understanding what nitrifiers are in your system and what it looks like when things are running well. And then when you start to see issues, do another sample, and then you can start to understand, what the differences are between when the system's running well, when the system's not running well. You can do a test before bioaugmentation and after bioaugmentation. One interesting thing we've found is that sometimes, we know the types of bacteria that we have in our nitrification product, the Nitrobacter and Nitrosomonas, and those are the kind of classical bacteria you see for nitrification that were first described in the literature. But in doing DNA testing, now we see a lot of different types of nitrifying bacteria. And what often happens is the bioaugmentation product that we put in is enough to get the system started. It's enough to help the recovery system. But then once the system's up and running, we might see those bacteria become a lesser population and see other types of bacteria handling the load during normal operations. And then when there's an upset, we see all those bacteria lower down. And then again, we repeat the process with an upset recovery dose, getting nitrifiers in the system, getting that process up and running, and then slowly over time, changing that population.
Thank you. Last one maybe for you. You mentioned EPS issues. Can you walk us through the early indicators that we should look for before EPS becomes a major problem?
Yeah, so one is on the microscopy. So if you're doing regular micro exams, you're going to visually see that change. And hopefully it's going to be a gradual change where you'll start to see it. Again, you want to do micro exams to your system regularly or have it sent out for analysis when things are going well. So you know what it looks like when things are going well. So you'll see EPS crop in if you're doing the India ink stain, or if you're just looking at it under phase microscopy, you'll see that. You'll also see issues in settling. So you'll see it in the settlability test, but also the SVI. If you're doing the sludge volume index calculation, you'll see that your sludge is taking up more space. You'll see it in the clarifier. You'll see it in the flock appearances. And then again, once you start to see those signs, look in your data for the other things and see if there are reasons why there might be more stress or toxicity or other issues that might be causing that EPS.
Well, thank you. I think we're right on time. Thank you all for participating. Thank you, Matt, for all this good content, and do not hesitate to follow up and write to us for follow-ups and specific case studies on your cases. Thank you, everyone.
Thanks, everybody. Bye-bye.
Bye-bye.